Okay, so in this presentation, we're going to go over the process known as translation. Well, here's a very simple diagram of a ribosome, and here comes a big strip of messenger RNA. What the ribosome will do, will read this strip of messenger RNA, and it will signal for transfer RNA to bring over an amino acid. For instance, in this example, methionine. And one by one, the ribosome will move down the notch of messenger RNA, signal for tRNA, transfer RNA to bring over an amino acid, in that example, proline. And one by one, the ribosome will move down a notch, signaling for tRNA to bring over an amino acid. And what is happening here is the ribosome is linking these amino acids into a big long chain. It's how a protein is created. And eventually the ribosome will come to a signal that says stop. Well, of course, I kind of just showed you a lot of the process before explaining anything. So let's go ahead and jump into this. So DNA and translation. If you recall the definition of a gene, it's a small piece of a DNA molecule that will code for the creation of a protein. In the picture, for instance, we have three genes. Gene A on top. For instance, gene A, you can see the bracket surrounding gene A. Perhaps that's the instructions to build a protein called hemoglobin. In the middle, you can see another small piece of DNA, gene B, and perhaps that's the instructions to build a protein called keratin. And so we have about 25,000 genes that make up human DNA. And the reason protein building is so important and just really a key fundamental area of biology is because proteins are used to build our cells, our tissues, our organs, organ systems, organism, and very much our body. You could argue that living organisms are protein making factories. So we need to understand how proteins are created. Proteins are created through a fairly complex process of two individual steps. Now, one we talked about in a previous video called transcription. And then when transcription is done, that's going to lead into today's topic called translation. Well, let's go ahead and review transcription just to keep it fresh on our head. So if we take a moment and review transcription, here's a strip of DNA. Transcription takes place in the nucleus because that's where DNA is found. If you recall, the double helix of the DNA is broken apart. My animated scissors represent the enzyme called RNA polymerase, and the RNA polymerase pries apart and breaks apart the double helix, the DNA. If you recall from the video that you may have watched earlier, is that transcription, what happens next in transcription, is that the messenger RNA nucleotides will match with the DNA. So look at the black letters on top, the A, the T, the T, the C, the C, the G on top. We're going to follow transcription along the top. Now, as we make RNA, that's what transcription makes. Transcription makes RNA. You'll notice that as RNA is being built, if there's a DNA A in black, RNA U will match. So as we do the process of transcription, we're building RNA. And in this case, we're going to build messenger RNA. And now that we've reached the end, Step 3, messenger RNA will detach and will move towards a ribosome. There it goes, moving towards a ribosome. Let's go follow that. There's the big strip of red messenger RNA that just exited from a moment ago, and it's about to go into the cytoplasm. So the messenger RNA will exit the nucleus. The messenger RNA will exit the nucleus and enter into the cytoplasm. And there it's going to find a cell part called a ribosome. Let's go look. So here's a ribosome, and here comes that strip of red messenger RNA. Well, once messenger RNA arrives at the ribosome, our next process will begin. The next process is called translation, and that's kind of what the rest of this video is going to discuss. So let's move on with our topic of today. So if you're in my biology class, I gave you one of these handouts, And this is called the genetic code. And what this is, it's a list of what we call messenger RNA codons. So what we're going to do is we're going to group messenger RNA into 3 nucleotides at a time, 3 letters at a time. For instance, you can see in the lower left-hand corner, GUG, guanine uracil guanine. That's a codon. Just above it, you see the codon GUA, guanine uracil adenine. That's another codon. And what these codons do is these codons are the code for amino acids. You can see in the diagram in the chart here the name valine and the name alanine and the name threonine. These are the names of the 20 different amino acids. If you count them all up, there'd be 20 of them. Starting in the top left, phenylalanine, that's 1. Beneath it is leucine, that's 2. Beneath that is isoleucine, that's 3. If you count them all, there's going to be 20 amino acids. And this chart is an organized diagram that shows which codon codes for which amino acid. In the lower left-hand corner, the codon GUG will code for the amino acid called valine, and you'll see how this all works in just a moment. And so what we're going to show you in a moment is that the ribosome is going to read messenger RNA 3 nucleotides at a time. The messenger RNA I should say, excuse me, the ribosome is going to read 1 codon at a time. 1 codon is made from 3 nucleotides. I want to point out a couple codons of significance. In the box flashing right now, you see the codon AUG. AUG is an important codon because this is the codon that really activates the ribosome and really begins this process. When the messenger RNA goes to a ribosome, AUG activates the ribosome and begins the process of translation. Well, so if there's a start codon, AUG, if AUG is a start codon, there's probably going to be a code to stop this process. And in the upper right-hand corner, you can see there's actually 3 of them. There's 3 what are called stop codons. So there's a codon to activate the ribosome, and there's a codon to deactivate the ribosome and basically let the ribosome know that the job is finished. So let's go ahead and look at an overview of this process to see how this works. If you have a copy of this genetic code diagram right here, it's going to be really helpful as we go through these notes. You can always Google the words genetic code to print one of these off if you're not in my biology class. So here's an overview of the process of translation, and this is the animation that we saw at the beginning on the title screen. So translation, if you recall, is the process for how a ribosome makes a protein. Proteins are created by the ribosome as the ribosome performs a process called translation, and this is an overview of that process. So step number 1, messenger RNA that was created through transcription enters the ribosome. And so in the animation, here we see that red strip of messenger RNA that we saw earlier. And notice how there's an AUG that has entered the ribosome. Step 2, the ribosome is going to read 1 codon at a time. Remember, a codon is made from 3 letters, 3 nucleotides. And so if you look on your genetic code chart, find AUG on your chart. AUG has the word start next to it. Not only does it have the word start next to it, but it's also the code for an amino acid. Transfer RNA will deliver the amino acid called methionine because AUG is the code for methionine. So here's tRNA, transfer RNA. That red cross-shaped object is transfer RNA, and it's carrying that orange circle labeled methionine. And so transfer RNA drops off methionine at the ribosome, and the ribosome really just repeats this process. The ribosome moves down a notch. The ribosome reads the codon CCA. If you look on your chart that you have, find CCA on your chart. It's the amino acid called proline. So CCA is the code for proline. So transfer RNA will deliver proline. Proline will link together with methionine that was already delivered. Transfer RNA has done its job, so it'll detach. The ribosome just repeats. It moves down a notch. The ribosome now encounters GGA, the codon GGA. If you look on your chart, look at GGA on your chart, and you find the amino acid called glycine. So transfer RNA will deliver glycine. And the process just keeps repeating. tRNA drops off glycine, and now we have a growing chain of amino acids being built. We have a protein being built. The ribosome moves down a notch to the fourth codon. And if you look in the picture, the fourth codon is UCG. If you look up UCG in your amino acid chart, in your genetic code chart, you find serine. UCG is the code for tRNA to deliver serine, and you can see that's just happened. So tRNA will now drop off serine. The ribosome moves down another notch, and the ribosome comes to the codon GUU. If you find GUU on your amino acid chart, on your genetic code chart, GUU is the codon for valine. So the ribosome is expecting valine. Transfer RNA will deliver valine. Transfer RNA will release valine, and the ribosome again moves down a notch. Well, now we come to something special here. Look at your chart. Find UAA on your chart. When you find UAA, that is a stop codon. That's the signal that the process is finished. The ribosome is done building this protein. And what happens is the ribosome actually breaks apart. The messenger RNA kind of exits and leaves because its job is finished. And what we're left with is this chain of 1, 2, 3, 4, 5 amino acids called a protein. Now, in reality, a protein is much larger than 5 amino acids. Proteins can be made from thousands and thousands of amino acids, but for simplicity, this one here was made from 5. Well, this was an overview. There's a few details I left out that I think are really important. I want to talk about those details now. Okay, so there's a few important details that I left out a moment ago in the overview that I really want to touch on. So first of all, some of these notes are going to kind of be repetitive of what we just said, but then we'll come to some new information. So translation begins when the mRNA codon AUG is read by a ribosome. So here's a ribosome, and here comes our strip of red messenger RNA. Now in this animation, notice that CCU is currently in the ribosome. The ribosome has yet to encounter an AUG codon. So in this case, that CCU is simply really just ignored. The ribosome hasn't been activated yet. And so one by one, the ribosome will simply move down and be reading these codons. And now the ribosome has encountered the AUG. And AUG is the start codon. And if you recall, if you look on your chart, you're going to need that chart again. If you look on your amino acid chart, the genetic code chart, find AUG. Transfer RNA is going to deliver over an amino acid. AUG is the start codon, yes, but AUG is also the codon for an amino acid. So tRNA, transfer RNA, will deliver methionine. Now, here's a big detail I left out a moment ago. We want methionine. AUG is the codon for methionine. That's the only amino acid that we want. If a different transfer RNA tried to bring in, for instance, leucine, that could cause a real big problem. So there has to be a way to prevent mistakes. Here comes transfer RNA. Notice it's carrying methionine as we want. Now, in this animation, I added 3 black nucleotides to the transfer RNA. I added UAC. Notice how the black UAC is a match. It's the opposite of the red AUG. We call the red messenger RNA letters, we call it the codon. Transfer RNA possesses what we call an anticodon. Anti is a prefix that means opposite. So what's opposite AUG is UAC. Transfer RNA in this example has the anticodon of UAC. That's a match. That means methionine will be delivered. So, therefore, the tRNA delivered methionine and has just exited. The ribosome will move down a notch. And now look what happened. Look at this. Transfer RNA is it's reading the red codon of CCA. If you look on your amino acid chart, the red codon CCA is proline. I'm supposed to have proline right now. But transfer RNA is trying to deliver histidine. This is an improper match. This is the wrong amino acid. The protein could be really affected negatively if this is allowed to proceed. So histidine in this example is the wrong amino acid. Again, the red codon CCA is the amino acid for proline. That's what I'm expecting. So because the red CCA codon does not match the black GUA anticodon, watch what happens. Transfer RNA will basically remove itself. Like the notes say, this prevents the delivery of the wrong amino acid. If you look at again CCA, I'm wanting proline. Here's the right tRNA. It has brought proline over. Notice how the red CCA codon matches the black GGU anticodon. That's a match. tRNA will deliver proline and exit. The ribosome moves down a notch. One by one, amino acids are linked together. Right now, the ribosome is reading GGA. Find GGA on your chart and you'll see that's the code for glycine. I want glycine and only glycine. Here comes tRNA carrying glycine. Perfect. Notice the red GGA codon. Notice the black CCU anticodon. That's a match. So therefore tRNA delivers glycine and one by one amino acids are linked together. The ribosome moves down a notch. Now the ribosome is reading the red UCG. Find UCG on your chart, and that's serine. I want serine, but look, transfer RNA is trying to deliver lysine. I don't want lysine. That's not the proper code. But notice how there's an improper match. The red UCG codon does not match the black UUU anticodon. Because of this improper match, the transfer RNA that is carrying lysine will be removed. That's perfect. We don't want lysine. UCG, I want serine. So the ribosome now is delivered serine by the proper by the proper transfer RNA. And so that's a match. And now the ribosome just keeps moving down a notch. Find GUU on your amino acid genetic code chart. GUU is valine. The ribosome wants valine. Here it comes. Transfer RNA is delivered valine. Notice how the red GUU codon and the black CAA anticodon, they're a match. So transfer RNA detaches. The ribosome moves down a notch. The ribosome comes to UAA. That's a stop codon. Notice there's even a few letters after the stop codon. There's a CAC after the stop codon. Well, that CAC really isn't going to amount to anything because the ribosome's been stopped. The ribosome will really just kind of break apart, and the mRNA, the red mRNA, will kind of just break apart and dissolve, and what is left is a protein. So translation builds a protein, but I really wanted to show you in detail because knowing the codon and anticodon is really important. It prevents the delivery of the And so here's a couple practice problems. If I were to give you the DNA letters TGA, pause the video and fill out the mRNA letters that TGA would create. I'm going to show the answer in 3, 2, 1. If the DNA letters are TGA, the messenger RNA codon would be ACU. Okay, again, pause the video if you like and try to figure out what is the tRNA anticodon. If the messenger RNA is ACU, the tRNA anticodon is UGA. Okay, and now here's where a lot of mistakes come on the practice problems. I have three groups of letters up here. I have DNA letters, I have mRNA letters, and I have tRNA letters. Which letters do we look up on that amino acid chart? Do I look up the TGA? Well, no, there are no T's on that chart. So I'm either looking up the messenger RNA or the transfer RNA. Well, let me ask you, what does the ribosome read? Does the ribosome read messenger RNA or does the ribosome read transfer RNA? I hope you know the ribosome reads messenger RNA. So you have to look up the messenger RNA. You look up ACU on your chart and you find the amino acid called threonine. A lot of problems, a lot of mistakes that students have made in the past come when they look up the transfer RNA anticodon by mistake. Here's another practice problem. Okay, in this practice problem, I've given you the messenger RNA of AAG. Pause the video. I'm going to go over the answers to all three of these boxes in 3, 2, 1. Okay, so to work backwards, the DNA letters TTC will lead to the creation of the mRNA codon AAG. Once I know the mRNA AAG, the tRNA is just the opposite. What's the opposite of AAG? UUC. And so now I have that same problem from a moment ago. Do I look up the AAG on the chart or do I look up the UUC on the chart? The ribosome reads messenger RNA. So you look up the messenger RNA, the AAG, and you find lysine. If you thought the answer was phenylalanine, that's because you accidentally looked up UUC. Here's one more. Okay, in this other practice problem here, pause the video, try to figure out the three boxes. I'm going to go over the answer in 3, 2, 1. So if I have you start with the tRNA anticodon, you have to figure out what messenger RNA codon would have coded for this. I hope you would come up with the letters GGU. And then you have to go backwards to the DNA. What DNA letters would have given rise to GGU? And that would be CCA. So again, now we have to figure out the amino acid. You have your chart in front of you. Remember, what does the ribosome read? The ribosome reads messenger RNA. You have to look up GGU, messenger RNA, and if you do, you get glycine. If you thought the answer was proline, that's because you accidentally looked up CCA. Here's another good practice problem. Pause this and try to answer these three questions. And so here's a big strip of messenger RNA that just entered that ribosome there. In order, try to answer these three questions. I'm going to go over the answers in 3, 2, 1. So the answer to number 1, list the amino acids that will be delivered to this ribosome. There they are, methionine, and then proline, and then serine, and then proline again, and then glycine, and then serine again. Let me explain. That first codon is a GAU. The ribosome will ignore that. AUG is the start codon. tRNA will deliver methionine. The ribosome simply moves down a notch and comes to CCG. CCG is the code for the amino acid proline, so proline is delivered. The ribosome moves down a notch. AGU, look on your chart, find AGU. Transfer RNA brings over serine. The ribosome moves down a notch. The ribosome is reading CCA. Find CCA on your chart. That's the code for the amino acid proline. And one by one, the ribosome moves down a notch, reading codons. GGA, look on your chart. GGA is the code for glycine. And the ribosome again moves down a notch. And now the ribosome comes to UCU, that's the code for serine. And eventually the ribosome gets to the end, UGA, look on your chart. UGA is a stop codon. And so the process is finished. Well, question number 2, what is the anticodon for each? The anticodon, again, are just the opposite of the codon. And so the opposite of AUG, the first anticodon, is UAC. So the anticodons are opposite of the codon. And question number 3, when finished, how many amino acids in size is this protein? We'll count them up. There are 6 amino acids. So there you go. If you're in my biology class, try these problems right here, and I'd be happy to check your answers, either before school or after school. So go ahead and pause the video and work these out. Good luck.